Okay, so for today's lesson, we're going to be going over the process of blastulation. So first of all, just to define some important terms, blastula is just a ball of cells, which will eventually develop into the gastrula. And then important differentiation to make between terms is polarity and differentiation. Polarity is the differences in region or distribution of parts of a cell. And this differences in distribution is based off mRNA, proteins, and pigment. And then differentiation is whenever it differentiates to develop its basic cellular function. So I think polarity has to do more with structure and differentiation has to do more with function. And then for just some normal physiological states of an animal cell, your resting membrane potential is going to be -70. And then you're going to have a higher concentration of potassium inside the cell, and then everything else has a higher concentration outside of the cell. So that sodium, that calcium, and that chloride. So the first organism that we're going to talk about with blastulation is going to be the sea urchin. And so here the sperm is going to penetrate your animal pole up here, and then that's going to start your different cleavages. So first you're going to have a vertical, then another vertical, and then a horizontal. So with these verticals, whenever you do those cleavages, it's going to have equal distribution in between. So those are going to be identical. But then once you reach your horizontal cleavage, here you can see the different distribution of proteins. So you're going to have different, and that's how it's going to eventually differentiate into these different blastomeres down here, which will eventually differentiate into your different germ layers of your cell. So here we have mesomeres, our macromeres, and then our micromeres. So for your micromeres down here, that's going to become the mesoderm. And then for your macromeres, that's going to become the endoderm. And how I think of this, I just think macromeres, endoderm, like endo, I don't know, I think like big butts. It's kind of weird, but I don't know, like big ends, macro with endoderm. And then your mesomere is going to go with your ectoderm. And then, while I remember, another thing I used to remember for micromere going with the mesoderm, I just think meso, like the mesoderm, meso middle, so the middle child, and then micromere, like micro small, so you think the middle child is going to get the least amount of love. Yeah, and then so eventually this mesoderm is going to become your muscle and organs, so just think meso, muscle. Your endoderm is going to become your gastric and respiratory tract, and then your ectoderm is going to become your skin. Now we're going to move on to the frog. So before this first cleavage happens, you're going to have your gray crescent form. And so how that happens is here you have your animal pole, and you can tell that just because it's darker, because it's more dense with proteins. So that's how you know this is the animal pole, and your sperm's going to penetrate here. And you have these two cortical cytoplasms, this lighter one and this darker one. And so whenever that sperm penetrates, you're going to get this 30-degree rotation in those cortical cytoplasms, and then they're going to mix. And so that's how your gray crescent forms over here. And again, that indicates your first cleavage is about to happen. So for the whole blastulation process for the frog, it's pretty similar to the sea urchin, just with a few differences. So again, you have your vertical, and then horizontal cleavage. The only thing is, compared to the sea urchin, the cleavages kind of happen simultaneously. They don't wait. With the sea urchin, you have your first cleavage fully happen, then your second cleavage. But here in the frog, your first cleavage is going to happen, but then before that one's even done, your second cleavage is going to happen, and so that's why it can differentiate a lot faster. And then again, here your blastocele is going to form, as same with the sea urchin. And then here's just a little map of what the endoderm, mesoderm, and ectoderm are all going to form from. And now for the chick blastulation process. This one is pretty different from all the others. So first off, just a little bit of anatomy. Here's your ovary, and then you have your oviduct, which consists of all these parts, the isthmus, the magnum, and the infundibulum. And then you have your uterus down here. So fertilization is going to happen in this infundibulum, and then it's going to go to the magnum, in which your blastodisc is actually formed. And we call it a blastodisc instead of like a blastula, just because it's a lot flatter than all the other organisms' blastulas. And then here you go to the isthmus, and that shell membrane is just going to get added on to that blastodisc, and then it'll finally go to the uterus, where that calcium carbonate shell is actually added. And then here, just showing the cleavages, you have a vertical, horizontal, vertical, and then it just keeps going until it differentiates. And then down here, just some important parts to point out. So you have your marginal zone here, which is in between your areopaca and your area pellucida here. So I think areopaca, opaque, that's where it's going to be very dense with proteins, and that's the outer layer. And then here's just another different view of it. Here you have your areopaca, like we just said, and then your area pellucida here. And again, here's just another view of that blastulation process in the chick cell. So here, this whole thing's called your blastoderm. You have the posterior marginal zone, and here's your epiblast, and then your areopaca down here. So what's going to happen is these hypoblasts are going to dissociate from this epiblast, and it's going to go and to form its own hypoblast layer beneath it. And that hypoblast, its function is just for support of the cell. And then this epiblast up here is what's going to eventually differentiate into those different cell types. And then here you have this little blastocele between the epiblast and your hypoblast. And I think that's all for this lesson. Again, we'll have our quiz on our Instagram for you to test your knowledge and understanding on the topics, and I hope you guys have a nice day.